Welcome to the conversation. I'm Paul Grondahl, the director of the New York State Writers Institute at the University at Albany. We're very excited today to have a faculty member and 3 students who are summer fellows, the summer fellowship program at the RNA Institute at the University at Albany. Now, COVID-19 disrupted many things, and I knew you would have been on campus doing hands-on lab work, but like everyone, you had to pivot to a remote learning model. So I also want to start with Dr. Andy Berglund, the director of the Berglund Lab and one of the faculty members overseeing this summer fellowship. Welcome, Andy.
Thanks, Paul. It's great to be here. I wish we were chatting face to face, but obviously we can't do that right now and with COVID-19.
Yes. So I'd also welcome Eva Clervoyant. She's a senior at the University at Albany, part of the project. Megan Barber at the University of New Mexico, and she's joining us from Albuquerque, New Mexico, in a different time zone and everything, which is the magic of Zoom, and Tyler Dion, a student at SUNY Geneseo. So why don't I ask, first of all, what were the challenges of running a lab when you can't actually bring the students to campus, Andy?
Yeah, you know, it was when we shut down the labs, it was hard because a lot of people still wanted to be in the lab doing research. One of the things we chose to do relatively quickly was start working on COVID-19. So a small part of my lab actually got back in the lab almost immediately. And this was through a collaboration with Upstate Medical Center that was collecting samples from patients, COVID-19 patients, and funded through SUNY. We started analyzing the RNA sequence from the patients and starting to think about, you know, how the virus was impacting the host, us, and also how it might interact with our microbiome. So we wanted to really start analyzing that virus. And that's how these students all got involved. And I should say, you know, these are 3 representative students, but there's 22 other students that are summer undergraduate fellows, and they're all working remotely to help us understand how this virus is impacting us.
That's great. So I'll start with Eva, because I know you've worked in the lab during your time at UAlbany as an undergraduate student. How different was it when you say you had COVID-19 samples? I don't know what that means. If you're working on a computer, are you looking at some kind of high resolution image of it, or are you just getting, I don't know, you don't get an actual sample by the FedEx delivery to your house, do you?
Actually, let me answer that, and then I'll let Eva jump in on sort of answering how it impacted or how she's been involved. Actually, Paul, so the first time one of my senior scientists drove over to Upstate and literally collected RNA that had been isolated from the virus. And then one of my other senior scientists turned that virus into something we call, basically we sequence the virus. So we're gonna sequence every nucleotide of this virus. It's almost 30,000 nucleotides long. And something that's, you know, we have different types of viruses. We have DNA and RNA viruses. COVID-19 is an RNA virus. That's partly why probably as the RNA Institute, we wanted to work on it.
Right.
And so, but yeah, actually we've also had them FedEx samples to us over from Syracuse, but they're not live virus. So the RNA has been inactivated. And then we actually, you know, basically perform some molecular biology experimental techniques so that we can then sequence the RNA. And so then the sequences of those viruses are sort of in the cloud or on the high performance computing cluster here at UAlbany. And then the students can access that data and help us analyze the data. And that's sort of where Eva, Tyler, and Megan and all the other students jumped in. And sorry for jumping in. I just wanted to provide some of the background.
So Eva, since you've worked in the lab with, I assume, you know, test tubes or beakers or whatever, I was never great at science, but how different is it than looking at a computer screen and just seeing a 2-dimensional representation of it, or 1-dimensional, I guess?
It's kind of cool in that when I do analyze the data from my experiments in the lab, you get to like see the like the computational analysis of it all. Is like it differs in terms of that, so after we perform the experiments, we see like the Excel sheets comparing the data from like different small molecules, which is in my case, and the difference in gene expression that they have. But when we did it this time around, it was kind of cooler in that we learned how to use different programs like RStudio, and we saw like graphs of it and things and whatnot, things that I didn't know how to do before. So I think the bioinformatics part of it was a little cooler than the physical science and the lab part.
Okay. So I keep hearing this term bioinformatics, and I'm a little bit hazy on what it means. I'll jump to Tyler. Can you explain that for somebody who's not a biology major like a lot of you are?
From what I understand, the best I can, bioinformatics is kind of looking at, well, biology, but like the statistics part of it, and just looking at different analysis. So like we're able to create graphs looking at differential expression. Yeah, I can't really think of any other way to explain
That's
it.
good. That's good. Megan, I read your bio on the RNA Institute website. Very interesting. You come from South Africa, and you said it was always a dream to come to a university in the United States. Why was that important to you?
I think because a lot of my childhood I visited the United States because my father's side of the family is American. And I remember my uncle bought me a Harvard sweatshirt, and I just always kind of was just enamored with the idea of these American universities, all the research going on, all of just how, high level the academia is. And I think it was just like a little girl's dream to come here, because I just didn't know my chances were better at getting higher education in the United States compared to South Africa. So yeah, that's kind of how I always felt.
Now, Andy, I know it's competitive, and I was happy to hear that you get a stipend, that you're paid for your time and work this summer, which is always nice. University of Oregon, University of Florida, very large, prestigious universities. Did you get the idea from them? But you mentioned you have some colleagues at those schools. Did you guys formulate this idea of this undergraduate RNA summer internship together, or how is there, three of them in the country?
So it was probably my past experiences at the University of Oregon, and to a certain extent University of Florida, that made me realize we could do something like this, where we could bring people from across the country together and to move forward our research. One of the key leaders in this group is Ryan Ming, who's our bioinformatics specialist. He actually received his undergraduate degree and master's degree from the University of Oregon. And he received his master's degree in a program I started back in 2011. And I started that program because, again, back then, we didn't have nearly as many people able to analyze the massive amounts of large data sets that were being generated in biology. So with some colleagues at the University of Oregon, we started this master's program. So in that case, it's an 18-month sort of intensive program where students learn to manipulate these very large data sets, analyze the data, sort of understand what's happening. So I realized we could take those skills that Ryan and others had and basically do it in a 10-week program this summer. We don't do everything they do at Oregon, and some of my previous trainees now run that program at Oregon. So I stay in touch with them, and actually they're the ones that recommended that I hire Ryan here at UAlbany. So it's a little bit of a long-winded answer to your question, Paul, but part of those experiences at Oregon and UF, we did a smaller program with some students, but it was more in person. It made me realize we could do this. And I just want to say I've been amazed how well it's worked. All the students, some of them had really no computer programming experience. I think might even been a little fearful about, what am I getting into? And I think everyone's really enjoying it, really learning a lot. I think getting to see, what the virus looks like at the single nucleotide level, saying, oh, look, this person has a virus. It is similar to everyone else, but it's a new virus, basically. I mean, they're all very similar, but the virus is mutating And so we can see what's happening in terms of the virus mutating in upstate New York compared to the city, compared to the rest of the country, compared to Europe, Asia. So the students actually analyzed the viruses from upstate and compared them to these other regions of the country.
Well, that's interesting. I'm also still a journalist. I write a weekly story for the Albany Times-Union. Can you give me some news, a news flash? Was there something different about the way it mutated in upstate versus other samples you looked at? Eva, did you find anything that was unusual to the samples you were looking at, or?
No, not exactly. I did analyze them on the single nucleotide level, but all of my results pretty much look the same.
Okay. Anyone else, Tyler or Megan, did you find anything that stood out that you, put a flag on?
Well, not really anything that stood out, but one thing we were able to do was we were able to create phylogenetic trees. And that way we could kind of like map where it has gone. And like it's like the contact tracing. You could use that to contact trace.
Yeah. So, I just keep reading it. It's really read as more of these stories a public health issue rather than a pure research trying to understand it. I keep hearing it's a very difficult virus to understand. It's tricky. And is that true? If you look at it, let's compare it to myotonic dystrophy. Is it more difficult to either understand or isolate or whatever than that RNA? You call them toxic RNAs before. Do they have any similarities when you're looking at the sequencing?
They're very different. I would say one of the things about this virus, which is COVID-19, is that, we've really only known about it for 7 months. So just to put it in perspective, for myotonic dystrophy, we've known about that toxic RNA since 1992 was when we first identified the mutation, and this mutation in the human genome that causes this form of muscular dystrophy, myotonic dystrophy. So, it's taken us many years in the myotonic dystrophy field to sort of understand what's happening at the molecular level and how that mutation causes that form of muscular dystrophy. With COVID-19, I would say it was early January when we first identified the sequence of the virus. So it's 2020, and we're still in 2020. So I think one of the differences between almost all other biomedical research is the rapid pace of COVID-19 research, also the public health aspect, sort of everything. So in some ways it's I don't know if tricky virus is the right word, but it is, we're learning a lot very rapidly, but it's also a little hard because it's like a fire hose. I mean, I don't know if you've done this yet, Paul, but it's sort of interesting. If you go to something called bioRxiv or medRxiv, there's websites that all the scientists are publishing their data, and there is just massive amounts of data being published.
Right.
And then also just like back to the sequencing, as Tyler and Eva said, we haven't learned a lot, but we are building on the general knowledge. So we do have these new sequences, but there's already over 60,000 sequences of this virus. And they're all similar, but they're all a little different. And that's what these phylogenetic trees tell us. And as Tyler mentioned, you can sort of trace back maybe where that virus is similar to. So, some of the viruses we have are very similar to others, say, sequenced from Wadsworth, upstate. Department of Health sequences. But some are sort of, maybe similar to some of the virus strains in Europe. So in a way, because there's so much data and we're adding to that database, but it is a little overwhelming. I think that's one of the hard things, good and bad, about this is so many of us are working on it, but all the data is just sort of, it's like a tidal wave of data, and sort of searching through that data, I'd say, is a challenge, not just for understanding the virus and sequencing it, but looking at all the information about the virus.
Right. Let's talk about, how you fit this into, college students like to have a summer break, maybe go on vacation, go to the beach or whatever. How did you fit this into your daily summer routine? Megan, what was this something, did you have to log on for certain classes or lectures, or you did it on your own time, or?
So
sure.
that
Yeah.
really thing about this was, so we did, for me, it was around 9 a.m. For people actually in Albany, it was 11 a.m. And so we'd meet, we'd kind of learn the techniques we needed to go forward and work on the data on our own. And that was great because then, yeah, we did go on our own. And then around 4 p.m. Eastern time, but for me here, 2 o'clock, you know, we'd meet in our smaller groups. And if we had any problems or questions, we were able to discuss it with our group leaders, with our fellow peers, which was really awesome because I feel like, you know, in my lab here, I kind of problem solve on my own, where this was a great opportunity where I had my free time, but I also had set times where I did things I needed to get done. And then I had a great community that if I ran into issues or problems, they were right there along the way, which is just kind of really awesome way to do research.
Yeah. Now, I guess Andy had mentioned there's another student from New Mexico. Do you know each other? It's kind of a random coincidence that there's 2 students from that state.
Yeah, so we do know each other. Levi is a really good friend of mine. We were actually part of the MARC program at UNM together. So it's just really cool. We found out about the program and, you know, we contacted each other and I was like, are you applying? And he's like, yeah. And I was like, cool, so am I. So yeah, it was just really fun. We were excited and
Yeah.
to represent New Mexico.
So here's a question for each of you. I was glad to hear that it was a fellowship with a stipend, and Andy had told me $3,000. How did you spend your $3,000, Tyler?
I haven't spent any yet, but because of this, I didn't have to work at a gas station this summer, so that's great.
That's better. I'd rather look at sequencing than pumping gas and stuff. Good choice.
Yeah,
Eva.
I was able to work at home and get my wisdom teeth out, and I only missed 1 meeting, and I was able to watch recording after, and I was just able to keep on working.
Nice. How about you, Eva? Will it go to something fun or to like textbooks or tuition or whatever?
I'm trying to save half of it, but I'm kind of a shopaholic, so I've been shopping mostly
All right.
to leave my house. Yeah.
How about you, Megan?
So I'm saving it, and as per suggestion of my program here in New Mexico, they suggested that we use it for when we apply to grad school programs. So that's kind of what I'm saving it towards. It's just to cover application fees.
So that's what I was going to ask. I know, Ava, you talked about medical school, but do you already have your, career path charted? Do you want to be bioinformatics specialist and work in RNA, or are you going to use this to go into another field? Or Tyler, what are you planning after you graduate?
Well, actually, before this program, I was kind of lost. I was just I knew I liked science, but I really didn't know what I wanted to do. I went to college wanting to be a doctor, but I just realized that wasn't for me. But because of this, I was able to see a lot of UAlbany staff or professors and stuff, and get to talk to them and interact with them, and I formed a lot of good connections, and it really made me want to go to Albany for my master's program in 2021.
Oh, congratulations. You'll be a Great Dane. All right. How about you, Megan? Do you have plans already after you graduate as an undergraduate? Or?
So my hope is that I will be able to get into a PhD program. And I am definitely, this program has helped me realize I love bioinformatics. My previous background is neuroscience, but it's kind of just made me realize moving forward, I do want to focus on more the bioinformatics. RNA, definitely, it's really interesting. I've spent a lot of my academic career learning about it, but this is the first time I really get to apply the knowledge I've learned in a more, I feel like in a workplace environment. So yeah, that's my hope. If the programs don't work out, maybe I'll do a post-bac research program. So, but definitely research is
UAlbany on your list of candidates for grad schools?
Definitely. It's
All right.
really, it's like top.
Excellent. You'll love the winters here. Anyway, Eva, how will this work in medical school? Are you on like a pre-med track at UAlbany, or what's your?
Yes, I'm a bio major, but I'm on the pre-med track. And research is just a great thing to be able to do. I think it helps in your knowledge of diseases when you are studying like the pathology and whatnot. So it'll definitely help me out in medical school. And I'm hoping that even after getting my MD, I can do some research on the side or do a PhD program if I decide to later on.
Okay. I still think of biology as, you know, dissecting frogs and things like this. It sounds like you're sort of computer scientists / mathematicians / statisticians. I mean, is there any hands-on with lab coats and Bunsen burners and stuff anymore? I guess that's chemistry, but I don't know, Eva, since you work there, can you do just as effectively remotely on a computer? Or do you need to be there getting your hands around, you know, scientific instruments and stuff?
I think I prefer to do it physically. Like I really enjoyed being in the lab and wearing the lab coat and doing the cDNA synthesis and whatnot and using the machines. But this is definitely another part of it that I enjoy as well.
Because I think of when I go to labs, I see all kinds of sophisticated equipment and high-powered microscopes and things. Is that, Andy, is that sort of what you've compressed those steps and then just given them the data, or you don't really need to do all that to analyze this toxic RNA, as you call it?
No, I mean, so a small part of the team was in the lab generating the data, and the large part of the team, these students included, were helping us analyze the data. It's in a perfect world, all these students would have been in the lab and collecting samples, turning it into the cDNA libraries, as Eva said, running on the sequencer, maybe doing some other sort of wet lab experiments and combining it with the computational biology or bioinformatics. So another way to think about this is that because of COVID-19, we chose to completely focus on a more computational way to analyze data, but you can't do that without the wet lab research as well. So really, you know, what you want to do is you want to do both. Like what I'd love for all these students to be able to do is say, okay, I'm going to isolate the DNA or RNA from, you know, whatever tissue sample we want or cells. I'm going to turn that into, isolate the RNA or DNA, synthesize the cDNA libraries, have it processed, be able to troubleshoot all of that, then be able to analyze the data, do the statistics, generate the analyses. So, you know, they were heavy on the back end, but in general, you really want to do it all. But, you know, for this summer, it didn't make sense. We couldn't bring in people in. So we have a small subset of people doing the hands-on work and a large group doing the analysis.
Okay. So I'm still trying to understand an analogy. It seems a little bit like you've got to find that grain of sand on the beach that has the, you know, what you're looking for. Or I try to read about Bitcoin and understand, and I can't. It seems similar, like blockchain is like this, a million numbers, and you got to find the one with the weird little, you know, original signature or something. Give me an analogy that I can get my head around. Are you scrolling through 8,000 screens and then you find one little, you know, blip on the thing that's what you're looking for? Or I don't know. Give me an analogy, Megan. How would you explain it to somebody that doesn't know what you do when you do this fellowship?
I would explain it as, I think one of the biggest things was we took the, you know, patient samples, and then we aligned it back to the human genome. And that's when we start looking, it's when you kind of compare one sample to another, and then you can kind of better like look for the differences. I'm trying to think of an analogy. I'm terrible with analogies, but I think it's just when kind of like a puzzle piece, you know, you can look maybe at one puzzle that has a similar you know, I'm terrible with analogies. I'm so
Is it at all like, I think of forensic, forensic work, people who worked at FBI or criminal justice, you're looking that fingerprint is a unique, are you looking through, a million fingerprints to find the one unique one that's different or something, or?
I wouldn't say like personally I'm going through. That's where the computational side really comes through. All these different programs we're creating does that for us, and it's going through, and then we get the chance to go look at that and like kind of see what the computers are helping us figure out might be like something unique, and then we go look, and then we kind of dive in deeper. So
How about Tyler or Eva? Do you have a analogy that you like to use? Tyler, go ahead.
Yeah, not necessarily an analogy, but it's not like we're looking through all the nucleotides. Instead, we simplify it by writing code to make the computer do it for us. So instead of doing something that takes years to find that 3 nucleotide sequence that you're talking about, it takes 10 hours, and we sit by and relax while it's doing it.
Oh,
and then
fun.
take the output, and then we are able to compare it and do whatever we want with the output. And that's where the real analysis comes in.
That's great. So I saw in your bio, and I've seen it mention R, just a capital R of code, which seems very mysterious. They don't even give it a whole name like HTML or whatever. It's like R. So, but you said you were a little rusty. How difficult is R? And explain it to me. And do all 3 of you code in R? What is R?
Yeah. So R is pretty much a coding software, but it's more for the analysis of biological things. So like for us, we would have the computer find that one grain of sample for us in each of those samples, and then we use R and load that data in and use it to wrap those grains in.
So even Megan, is R difficult? I mean, it's kind of like you have to learn a foreign language in 10 weeks or something, or?
yes, but it's really, well, I guess it's like a foreign language. The more you kind of just throw yourself in and you just kind of expose yourself, you just pick it up. But it's before I was doing things that week 1 was so difficult, and I was like, I don't know what this stands for. But you just kind of start to realize like, oh no, I know what this is, and it just comes really naturally, to you eventually, which is like a really cool feeling. It's a cool sensation.
Yeah. How are you at R, Eva?
I'm okay. I don't think I'm very tech savvy, but I'm definitely learning. And I think this is just a really good program because there's so much support. Like if I don't understand something, I'll literally message Megan or my team leaders or even Ryan, and everyone can help you out. And sometimes it helps to make a cheat sheet because sometimes I don't remember what every code does, but when you do remember off the top of your head, it's like oh my God, I remember this. It's like picking up a new language, exactly. Yeah.
Excellent. So how will each of you mark the end of this? You still have a paper to do, right? Are you procrastinators, or are you ready if I wanted to quiz you today on what you're going to present? Are you pretty well along with yours, Tyler?
I would say yes, but I just ran into a lot of issues doing it, but I'm definitely not a procrastinator. I'm gearing up, and now that my data working properly, I aligned it the right way. When you're trying to put $1 in the like one of the self-checkout things at Walmart and it's not going in,
Right.
I got it to go in, so now I can check out.
Nice. How about you, Megan? What can you explain what you'll be presenting on or writing about?
So I'm very much like Tyler as well. Like once I get the ball rolling, like it's good. But I am kind of looking at myotonic dystrophy and how it's affecting the central nervous system. So I am kind of, utilizing data from another paper, and hopefully I'm just hoping to go through and maybe spot something that stands out. But my goal is to kind of just maybe understand how the CNS is being affected, and hopefully I can present on that.
Okay. How about you, Eva? Are you looking at myotonic dystrophy or COVID-19 or?
Looking at myotonic dystrophy, I'm focusing more on the small molecules and how they affect gene expression and splicing. I'm looking at two small molecules in particular based on some photos. I mean articles.
That's okay. We're having a couple glitches. That's technology, you know. We'll have to get somebody to reprogram this in R later. So we're kind of coming up towards the end of our time. I'm wondering what I didn't ask Andy. What should we add here so people watching this, you know, would have a different level of understanding. These students must be pretty dedicated. I know other college students that are kind of slacking for this summer, but this is, would you say this is harder than a general class, or more work, or more intense?
Yeah, I would say I've been really pleased how engaged all the students have been. Every day they've engaged with each other, with our group leaders, and also they're, you know, wanting to learn about things. You know, all the questions they ask. You know, one of the things we did early on, it was probably the first week or second week, you know, I introduced the whole group to a family that has myotonic dystrophy. And the family was generous enough to do a Zoom meeting. And I think we ended up like a 2-hour meeting. And all the students and everyone was asking questions. And I think what was interesting for the students is that 2 of the family members that have myotonic dystrophy are college aged. So they could really relate to each other. And I think that helped, at least for myotonic dystrophy, you know, frame the research for them to understand how it impacts people. And I think that really sort of motivated them for that portion. Mm-hmm. And then the 3 students you happen to be talking to all are associated with my lab. So they're now studying myotonic dystrophy again for the final project. You know, I think for COVID-19, you know, maybe they don't know anyone personally, but it's just having such an impact, obviously, on the world, that getting to do something, I think, really motivated them. So I must admit, in the very beginning, I was slightly worried that, you know, I'm not seeing these students. You know, some of them I may, you know, like Tyler and Megan, I've never even met in person yet. But so I was a little worried that maybe we wouldn't pull together as a group and, you know, they might sort of drift away, or it's just hard to know when everything's remote and when you haven't actually even met people. You know, are they going to really engage? Are they going to get a lot out of this? And I've been thrilled. I feel like everyone is really, you know, I said I'd like to see 20 hours a week of sort of work, research, and I think everyone's done that easily. So I'm thrilled with the outcome.
That's great. Any final words, Eva? How long is, I mean, many years to get your MD, right? You looking at 4 years beyond this or more? Or?
Yeah, another 4 years of medical school, and then like 4 years of residency and fellowship.
Wow, are you prepared for that?
Yeah, I'm excited.
Excellent.
It'll go by fast, hopefully.
And even you were talking of PhD program, Megan. Is that also, you're looking at several years? You know.
Yeah, so I'm hoping I'll be able to do my PhD within 5 years, and then I definitely would like to do a postdoc for 2-3 years, just kind of widen my horizons, go see, you know, what other new techniques I can learn.
Excellent. And Tyler, you mentioned you were kind of lost when you started, like what direction, what career. You think you found something now where you could see yourself making a career out of this?
Yeah, definitely with research, because when you're sitting in class, all you can really think of with all like these facts and stuff is just like science is done. But now doing this stuff and seeing COVID-19, these people who have a disease, you see that it's real time and it's ongoing, and just something as simple as RNA can really be complex. It's be a whole world of research. And it really has excited me and wanted me to do some more research.
Excellent. So this has been really interesting. I want to thank our RNA summer fellowship recipients. It's a competitive program. They're paid, and it sounds like some of you are looking at, or in Tyler's case, enrolled in the graduate program at UAlbany. That's good to hear. So we've been happy to have on the conversation today Tyler Dion, Eva Clervoyant, Megan Barber, and Dr. Andy Berglund, the director of the Berglund Lab and a key member of the RNA Institute at University of Albany. Thanks for joining us on the conversation. You can see this archived interview and others on our YouTube page, NYS Writers Institute, our website, nyswritersinstitute.org. And we'll also give you a shout out on Twitter, Facebook, and Instagram. Thank you all from here from all over, from New Mexico, from Rockland County, from Albany, and I'm in Guilderland. So thank you all.